Hello guys, welcome to my channel Zoologist AK, and I am Aarti Kaushik again here with another topic, that is ELISA enzyme linked immunosorbent assay. This method replaces RIA, that is radioimmunoassay. So in this radioimmunoassay we used a radioactive labeled molecule, but in case of ELISA we use an enzyme. Here we use two enzymes particularly. First one is our alkaline phosphatase, and second one is horseradish peroxidase. So let's understand what this ELISA method is. The method involves the binding of an antibody molecule to the walls of a plastic tube, and these plastic tube consist about 96 wells to which we bound this antibody molecule. Then the test and labeled antigens are added, which will compete for the available antibody for their binding. After a prescribed reaction time, the tube is washed to remove all the reagent except those which are bound to the antibody molecule. The amount of enzyme labeled antigen, this antigen is enzyme labeled, which will bound to a particular antibody molecule. For the reaction in between the enzyme that is labeled to this antigen and the substrate that we are going to add into the plate or test tube will determine the proportion, or will determine the reaction between antigen and antibody molecules. So here the amount of product formed is proportional to the amount of enzyme present and inversely proportional to the amount of test antigen. And this enzyme activity between the substrate molecule and the enzyme that is labeled to the antigen molecules is measured through optical absorbance, but fluorescence or chemiluminescence techniques can also be used here. So before understanding this process and its type and application, we need to understand the basic principle on which this ELISA method is based. So in an ELISA assay, antigen is immobilized to a solid surface. This is done either directly or via the use of a capture antibody itself. Here we can see this is our captured antibody molecules, and this is our defection antibody and target protein. So here this antigen is immobilized with the help of these captured antibody molecules. The antigen is then complexed to a detection antibody that is present in here and shown by the green color. This detection antibody is conjugated with a molecule for detection purpose, like it is here it is labeled with biotin and streptavidin, HRP, enzyme, or any fluorophore compound. An ELISA assay is typically performed in a multi-well plate. As we know, it consists 96 to 384 wells, which provide the solid surface to immobilize this antigen molecule that we call here target protein. Immobilization of analytes are something that we are going to analyze. So basically these are antigens or protein molecules that will facilitate the separation of the antigen from the rest of the component in the sample. These characteristics make ELISA one of the easiest assay to perform on multiple samples simultaneously, because it differentiates different protein molecules from one another. Let's understand the ELISA with this picture. Here we can see that we used SDS PAGE gel electrophoresis. If you don't know what is this, sodium dodecyl sulfate polyacrylamide gel electrophoresis, then I am going to share the link in the description box given below. You can click that link and watch that video for clear understanding. So here we are separating our protein molecules. So here the loading and separation of protein will occur. After that we will transfer it to a nitrocellulose membrane, and we will block the membrane with neutral protein like BSA and milk, BSA bovine serum albumin, that are used to block the membrane. Then further we will incubate the membrane with primary antibody molecules that are specific to that particular protein. Then we will wash it for the removal of excess antibody molecules that are not bound in bounded state. Further we will incubate the membrane with HRP labeled secondary antibody. So here this HRP labeled secondary antibody, HRP is a horseradish peroxidase, that is an enzyme. It is used for the detection purpose, that, because whenever we will add a substrate molecule for this particular enzyme, illuminescence will occur and we can detect the enzyme present in the sample or in our blot, so that we can identify how much product is formed. So here we will incubate the blot further with chemiluminescent HRP. Chemiluminescent HRP means horseradish peroxidate substrate, that will be specific substrate for this enzyme, and we will expose it to the film for the detection purpose.